Hello, everyone, and welcome to today's live broadcast, Introduction to LabCoat Live SmartStart Training Courses for Expi Protein Expression Systems, presented by Stephanie Culbertson, a technical training specialist at Thermo Fisher Scientific. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots, sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit at www.thermofisher.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen, type the questions into the drop-down box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, please use the Ask a Question box and let us know you're having a problem. Please join me now in welcoming Stephanie Culbertson, our presenter. I will now turn the presentation over to her. Welcome, Stephanie.
Thanks so much, Susie. Appreciate it. And hello, everyone. Thanks for joining the session today. So my presentations may be a little bit different than some of the other ones that you've been watching today. I'm going to talk, not a technical presentation, but actually talk to you about training support that Thermo Fisher can potentially provide to you or someone you know to help you get started with using the Expi protein expression system. Many people may not be aware that Thermo Fisher's Life Science Group offers education services for our customers, or you may know of some things we offer but not others. For quite some time, we have offered training related to things like our applied biosystems instruments. If you buy a qPCR sequencing machine, one of our field application scientists will come out to your lab to help you get started using it. But in the past few years, we have expanded our goals with training, and we want to cover broader workflows and more applications that we can help you with. So one of the areas that we have expanded to in just this past year is protein expression and helping people get started using the Expi protein expression systems, the ExpiCHO, Expi293, and the brand new ExpiSf insect expression system that just launched a few weeks ago. So if that sounds like you, the training may be helpful. And I'm going to go tell you a little bit about what we offer here. Besides just trying to expand the application technical areas that we cover in our training from Thermo Fisher, we've also been trying to think about different ways that we could provide training and help more people with education on the products that they're using. So of course, we have added a few more classes that our field application scientists can offer at your lab. We do have several courses that you can take coming into training center labs that we have at some of our Thermo Fisher sites, where we'll do hands-on training in a lab with you and classroom training. But as I said, we've been looking at ways we want to reach out to even more people. And so we have this new offering that we're calling LabCoat Live, which is an online training. But it's more than just your basic webinar. So I'll begin the talk telling you a little bit about what LabCoat Live is and how it differs from other online training you might be familiar with. And then I'll tell you go into a little bit of detail on what we actually cover in our protein expression courses covering ExpiCHO, Expi293, and ExpiSf. So what is LabCoat Live? So the idea behind the training for us was really to try to replicate the experience that you would have if you came to one of our training or customer experience center labs, where we'd be doing classroom and hands-on lab work together in our labs, but set up that same type of training from the convenience of your own lab. So what we'll do is actually send you reagents, send you a kit, and some specific protocols that we want you to use to do an experiment with that kit. And then we'll kind of do a virtual version of those classroom and lab interactions. So we'll have interactive online lectures. Again, it's not a big webinar where there's hundreds of anonymous people. It's interactive. They're small classes. We right now limit to maximum of 10 people, and we encourage you to participate. You can talk to your instructors. You can ask them questions. You can share data with them to get feedback on how your experiment is going so far. So it's really as close as we can replicate to what you would get with an in-person training. Then, of course, we're following up lectures with your actual hands-on experiment that you'll be doing in your lab. But to support you, since your trainer is not there with you, there's actually a dedicated class website that you can go to that has copies of those lab protocols, the slide deck handouts to refer back from the lectures, and also, whenever possible, we provide videos showing showing the instructor doing the techniques in the lab and showing particular steps in the workflow that are really important to do the technique correctly, so you can see it there. So it's almost like having somebody there with you. We'll recreate it as best we can. And then, of course, we'll follow up after each major step of the experiment with another interactive session where you can share your data. Your instructor will actually look at it with you and talk about what happened, how it went, and any tips that can help you improve in the future. In the case of the Expi systems as well, these are more complex workflows. As you all know, they take multiple weeks. So we actually start this process again. We go through this cycle multiple times where we'll have a follow-up to go over results together, and then we'll have a lecture going over the next step in the workflow that you're going to do in the lab. You go and do those steps, and then we meet again to go over your results, and then at the end have a final review of everything that you did there. So we do our best to recreate what you would get if an instructor actually was there with you. So how does this work? So the online lectures are provided through a system called GoToTraining. It's somewhat similar to what we're using today for this broadcast, or if you've used something like WebEx or Live Meeting. But GoToTraining has some nice interactive tools, so definitely allows audio. You'll either be there, you can speak to your instructor if you want to, or you can also use a chat box to interact during the sessions. We have even drawing controls where we can give you control to highlight things on a screen if we're trying to point something out in an image, for example. And there's reference files you can download directly during that lecture. So lots of tools that you can interact with your instructor as they're presenting. Then I mentioned that dedicated class website that you can go to get all your information. So these websites are actually hosted on the Thermo Fisher cloud. If you go to our website, thermofisher.com, and click on the cloud icon, we've got multiple applications there, various things that can help support you in your research. And one of them is now an app called Education Connect, which is where all of these class websites are hosted. The class websites look something like this. So it's a dedicated page for each class and each session of the class, not even just the course in general. Again, these are all resources, copies of the slide decks, those videos I mentioned that'll help you with techniques. All of this is accessible from this page. And another feature on this page that I didn't mention earlier is a dedicated discussion forum for the class. So you can post any questions you have or updates for your instructors on your experiment and how things are going. Whenever you post to this discussion forum, the instructors will automatically get an email informing them that you have posted a message, and they will get back to you quickly. Perhaps not immediately. Everybody's going to be working on different things in between the online sessions, but they will get back to you probably within a few hours, and this is really your dedicated kind of technical support forum for the few weeks that you're in our class. So it's a nice resource to have there. So before I get into the specifics of the Expi expression classes, I thought I'd pause for a moment. Susie, do we have any questions so far about how LabCoat Live works in general?
We don't have any questions quite yet, but I know our live audience will bring us some for the end.
Sounds good. Great, let's move on then, and we'll talk about the specific classes that we offer for protein expression. And there are classes for other application areas I should mention, but obviously for ExpressionWorld, we definitely want to focus on these. So the main ones that you all might be interested in, so again, we offer these classes that we call SmartStart with a goal to help anyone who's new to the systems getting started, or maybe if you already worked with the systems a bit, but you feel like you could optimize your results a little bit more, we'll help you get started with the system, this SmartStart training. The courses for ExpiCHO and Expi293 are quite similar. In both cases, the courses run about 4 weeks. It's actually 4 weeks plus 1 day for a final session at the end. In both cases, we'll send you the main expression system kit or the starter kit that we have established for the systems, either ExpiCHO or Expi293. That has all the key reagents that are specific to these workflows. We will ask you to provide some equipment. We can't provide everything that you need to do expression, so we will, of course, assume that you have a basic cell culture lab set up, that you have your incubator and your shaker all ready to go. And we do actually ask that you provide a few of the consumables specific to this workflow. So things like flasks or pipettes, things like that. We do have a checklist of what those things are that you need to have on hand before the class starts, and we will send that to you before you get started. Of course, for the online sessions too, you'll need a computer with internet access. There aren't any official prerequisites for the class, but just to give you a little bit of knowledge, these are definitely introductory classes with regard to these specific products and this specific application. But these are probably overall maybe intermediate level classes. We do assume that people joining this have at least some background knowledge of basic cell culture techniques and basic molecular biology techniques like cloning. We'll cover them very briefly in the class, but our main focus in these courses is really about the things involved in the system kits, the cell culture, and the transfection steps that are the most important for the protein production process. So just want to set some expectations there that we do assume a little bit of background knowledge coming into this, but somebody who at least has some knowledge of protein expression would do fine coming in here, even if they haven't done it before. The ExpiSf system class is a little bit longer than the other two. Most of the other aspects are the same. Again, we're going to provide you with the system kit. That's the basic starter reagents for the system. Same equipment required, same prerequisite requirements as the other courses. The main difference and the reason why this class is a little bit longer is just that the ExpiSf system is a baculovirus-based expression system. And so you need a little bit longer to add those steps to make virus before and do infection, as well as doing transfection and the other things that are involved. So a little bit more complex workflow, and we want to spend a little more time with you. In both of these cases, too, you'll note if you're really working on your own, working with these products would actually probably go a little bit faster. So we've added a little bit of time at some of the key workflow steps to make sure that if something goes wrong, you have some time to do another run of get another cell culture started or do another run of the experiment. So we try to build in a little bit of buffer time in case you need a little extra time to redo one of the key steps. The learning objectives for all the courses are pretty similar. So again, I think I mentioned our target audience for these classes are really scientists with some background experience, and maybe you've done E. coli expression, but you're new to doing insect or mammalian expression. So anybody who's getting started with these kits, or maybe you've tried it once before and you need some help optimizing, our goal with this is to help you. So we'll definitely be outlining the workflow and highlighting for you what we think are the most critical steps that you need to pay the most attention to. We'll make sure you understand all the components of the kit, especially the ones that are unique to the system, like the enhancers and feeds for the protein expression process, and why they're in there and how they're going to help you to use them. And we'll really go over the fundamentals of the other steps that you're more familiar with, but some of the things that people kind of take for granted. For instance, a lot of people focus on the cloning and the transfection, but they kind of take for granted the cell health. And so we'll really focus on how to properly subculture and keep your cells as healthy as possible as a really key part of the workflow. For the ExpiSf system, we will also, of course, cover how to make the BACMID and how to efficiently produce baculovirus to do cell infection. And we'll go over the proper protein harvest in all cases. And at the end, we hope that you'll leave knowing, again, those key steps throughout the workflow and have a good idea of which steps you can optimize to most improve your protein yields. So for the ExpiCHO and Expi293 courses, here's a little bit more information about how those courses actually look. So and during those 4 weeks, there's actually 4 of those interactive online sessions. We've broken down the workflow high level, basically into thawing and subculturing your cells, doing the transfection and adding the enhancers, and then actually harvesting your protein and detecting and quantifying your yields. So to support those major steps, we'll start with the online session, just overviewing the kits and the components and really focusing on the details of the thaw and subculture. And then the goal will be for you to go into the lab and actually perform the subculture and have the cells go through a couple of passages. Then we'll meet again to discuss transfection right before your cells should be ready to do the transfection in the lab. And then right after the class, you'd go do the transfection in the lab, hopefully. And then same thing, as your cells are producing their protein during the protein expression period, we'll meet again just a few days before your harvest, go over concepts that should help you in the harvest. and then have you go do that. And then at the end, we'll have a final summary, a final review, go over the results that you've seen, any questions that you have on things that you saw, and any tips we have to optimize for the future. The 4 sessions are each 60 to 90 minutes. The first 2 sessions are a little bit longer. You can imagine in the first session, not only are we covering the technical aspects, but also we want to set expectations for the class, tell you how LabCoat Live is going to work, what the schedule is like for the class, make sure everybody knows when the next online session is, things like that. So that session and the transfection are a little bit longer, and the last 2 sessions are usually about an hour. Those 4 sessions are delivered, as I mentioned earlier, over 4 weeks plus 1 day. So this is kind of how it breaks down. So we'll have the initial session, and then you have about 2 weeks for your hands-on time of thawing and subculturing your cells. We'll be checking in with you online in the discussion forum to see how things are going in those in-between periods. And then again, we'll have a full online interactive lecture and discussion session right before your cells should be ready to do the transfection step. Then have you do the transfection and meet again right before protein harvest, and then have that final results and review once you should have had time to do your protein analysis and see how things went. Any questions so far on these classes, Susan?
There are no questions so far.
Great, thanks. All right, let's look at the ExpiSf, which is quite similar, but as I mentioned, it's a little bit more complex workflow, since in addition to all those steps of working with the cells and your optimal protein yields, we also have to make a virus, make a baculovirus stock. So it takes a little more time, and we do have 1 extra online session for that class. So adding on to the kind of workflow breakdown before, in addition to the subculturing and thaw of your cells in the first session, we'll also be talking about doing the transformation and isolation of your BACMID and how to screen for good BACMID clones. The next session we'll discuss how to actually transfect the BACMID and how you're going to harvest and titer your baculovirus stock. Then the third session we'll talk about actually doing the infection of the cells with that baculovirus stock. And then the last 2 sessions are very similar to the ones for ExpiCHO and Expi293, where we'll talk about just your protein harvest and tips for efficient harvest and detection, and also go over that final review and optimization tips. And again, each of these sessions is between 60 and 90 minutes, depending on the subject. The earlier ones usually being the longer ones. But our goal is definitely to make sure that everyone, again, can be interactive and ask the questions, and that we can help each individual as much as possible. Here's that calendar breakdown of how those sessions are going to look. So a little bit longer, everything's stretched out. Like I mentioned, we add a little bit of buffer time where we can, because we want to make sure that you have plenty of time to do the analysis and production that you need, and maybe even a little extra time in case you need to repeat some things. So in the case for this class, we give you about 2 weeks, a little bit over 2 weeks, to get that, not only get your cells, your ExpiSf 9 cells thawed, but also produce your BACMID and maybe even repeat transformation and screening of BACMID if you need to, just in case. Then another 2 weeks to complete your transfection and harvest your baculovirus and do the proper titering that we recommend to really get the best possible results. But once you've got your good baculovirus stock, then the actual protein production goes very quickly, and it only takes a few days to produce protein, harvest, and finish up with our last online lesson. So packaged within 6 weeks there. So that's really it, high level, but hopefully that helps you know if these classes might apply to you or someone that you know that's going to be working with these systems or might need some help. So if you're interested in the, let me get back here. So if you're interested in the classes, this is the webpage that you can go to get more information is thermofisher.com/labcoatlive. When you get to this page, there's links for each of the classes to another page that has a lot more detail on similar things that I just covered, what's involved in each class, what's covered, what comes with it. You can also, the pricing for each class is on this page, and you can also see the dates that upcoming courses are offered. And then you can submit a registration, and we'll get back to you on class availability and all of those things. If you have any questions, I apologize that I didn't put it on this slide, but we have an email address that is listed on this LabCoat Live page. It's just labcoatlive@thermofisher.com. You're welcome to send us questions anytime as well.
Thank you, Stephanie, for that informative presentation. And we will now start that live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click that Send button. We'll answer as many questions as we have time for. So let's take a look at our incoming questions, and we do have a few already coming in.
Great.
The first one is, what is the cost of the classes?
So the actual cost, it does vary by class, so I won't spend time listing it out here, but let me take us back to that slide with that website that you can go to. So all the class prices are listed at thermofisher.com/labcoatlive. The pricing varies by course based on factors like the relative cost of the reagent kits that we're sending you and the number of hours of online training and consulting time that you'll have with the instructors. One thing that's not listed on this page, and again, I'm sorry I didn't put it on a slide, I didn't think of it ahead of time, but another site that you might want to go to, we do actually offer some discounts, like group discounts, if maybe multiple people from your lab might want to take a class, or if you sign up for multiple courses or refer someone else to one of our courses. For that, there's another website you can go to, and I'll try to say it slowly so you could maybe write it down. It's learn.thermofisher.com/promos, PROMOS. In promotions. And at that site, again, learn.thermofisher.com/promos, you can find out more about those group discounts potentially. That might help some people.
Thank you, Stephanie. And is there any training on RNA sequence?
For LabCoat Live specifically, there's only one class that we're offering on sequencing, and that one's related to analyzing sequence of cells, but that's all on that LabCoat Live page. We do have other sequencing instruction available. I don't think we have a specific kind of scripted class on RNA-seq, but we definitely have field application scientists that can help with RNA-seq. So if you're interested in training on that, I encourage you to contact another email address, and again, I'll try to say it slowly, but it should be pretty easy. It's customertraining@thermofisher.com. If you reach out there with your interest in RNA-seq training, we can confer with our field application scientists in that area and see what they can provide for you.
Thank you, Stephanie, and thank you to our live audience. We have some great questions coming in now. What would happen if I have a problem with my experiment and fall behind the class schedule?
Very good question. That's something that I'm sure people might be concerned about, but as I did mention, we did try to build in some buffer time so that some steps could be repeated where we know things like, maybe you thaw your cells and they don't seem to be recovering well. We did build in a little bit of time where you really could thaw some more cells. But if we do run into a worst case scenario where something bad happens and we really can't recover for it, we'll definitely work with you on the best resolution. We can't fully refund the classes once we send you the kit. A lot of the pricing of the classes is based on the reagent kit that we are sending you, but we can definitely reschedule you to another session, or we'll find alternate solutions. We generally have recordings available of the online sessions that we have, and we can provide those to you. So.
Thank you, Stephanie. And our audience members want to know, what if I have to miss one of the online sessions?
Because I accidentally already answered that question. So if I just mentioned that we generally do try to record the sessions for that exact case in case someone is unable to miss the class, or sorry, unable to get to that online session. Or if we haven't recorded that exact session, we usually have previous recordings of the same class, and you can just contact us. And again, that general email address that you can send questions to is labcoatlive@thermofisher.com. If that happens during your class, then one of our class coordinators or the instructors will make sure that you get access to the proper recording.
Thank you, Stephanie. And who are the instructors? It's actually a two-part question. Who are the instructors, and do they work in research and development?
So our instructors are all very experienced scientists who have worked on applications similar to this. Most of them actually work now in our support functions. So they're either field application scientists or technical support scientists who spend their time answering questions on these products every single day for people that are out there using them. Every once in a while, R&D scientists will be a trainer for us, but most of the time, their primary work has to be in the lab, but they are definitely involved in the creation of all of our course content. So they are involved, at least indirectly, and the support scientists who are your trainers are very experienced scientists and are able to give you a lot of tips that will help you.
Thank you, Stephanie. And I want to remind our audience that any questions not answered today will be answered via email. Stephanie, would you like to provide our audience members with a closing remark before we head out today?
Not much new to say, just definitely, again, our main goal is wanting to support people and help them be successful, hopefully the very first time that they use these products if they're just getting started using them, or help people to be successful. That's our main goal in general in technical support and across Thermo Fisher. We want your research to be successful, and that makes us successful too. So yeah, this is just one more way, a little even more proactive than just troubleshooting questions, where we can hopefully help you before you even run into any issues. So definitely encourage you to sign up if you're thinking about it, and we'd love to see you in the classes.
Thank you again, Stephanie. And I also want to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through May of 2019. Please share that with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We'd hope to see you again soon. Bye-bye.